Hello everyone, let's get started. Today we're going to be discussing cellular respiration and fermentation. To review, we've been talking about energy and how energy flows in living cells. We've discussed how endergonic reactions are paired with exergonic reactions through this sort of a middleman molecule called ATP. We talked about how ATP is used by a cell by constantly breaking off its outer phosphate group and releasing a bundle of energy, and then putting that phosphate group back on and saving or storing a little bit of energy. The energy that is released from exergonic reactions, in other words, is captured by taking ADP and a phosphate and that energy and rebuilding ATP. Then, when energy is required for an endergonic reaction, that energy is provided by breaking the phosphate group back off. ATP is brought to ADP plus a phosphate group plus a little bundle of energy. Remember, we called this energetic coupling. The ATP that's needed for energetic coupling to occur in a living cell is made through the processes that we're going to be discussing today: cellular respiration and oxidative phosphorylation. Now, we'll be talking about these metabolic processes for aerobic organisms primarily. So organisms like human beings that use oxygen and perform cellular respiration and oxidative phosphorylation aerobically. When I use the word cellular respiration, I'm talking about the process that oxidizes glucose to carbon dioxide, and along the way collects all of the electrons that get released from the covalent bonds in glucose, and then harvesting the energy from those electrons. And when we talk about oxidative phosphorylation, we're talking about a process that uses a hydrogen ion gradient and a very important molecule called ATP synthase to make large quantities of ATP for use in the cell. So, cellular respiration is actually a long series of chemical reactions. It's actually grouped into several sets of reactions. The first is glycolysis, then there's pyruvate processing, the Krebs cycle, and finally electron transport. Oxidative phosphorylation is going to use the energy that is harvested during cellular respiration in the form of an ion gradient, a hydrogen ion gradient, to build ATP using the enzyme ATP synthase. Let's take a minute and review some of the key features of this molecule ATP. Remember, ATP stands for adenosine triphosphate. And this is a small molecule that has a high potential energy because it has 3 phosphate groups attached to it. And you can see those in the drawing on the right-hand side. Here's the sugar, here's a nitrogenous base, and here are 3 phosphate groups. The highlighted area shows the bond that will be broken when ATP is split into ADP plus that phosphate group, plus a little bundle of energy. Now, ATP is used by breaking it and building it, and breaking it and building it over and over again. When we break the phosphate group off, energy gets released that will fuel endergonic reactions in the cell. When exergonic reactions occur, This phosphate group will be put back on, and that energy that was released by the exergonic reaction will be temporarily stored in that bond. Remember that when phosphate groups come off of ADP, phosphate groups can be put right back on to make ATP, but they also have other uses in the cell, and primarily the other uses are to regulate the activity of other proteins, including other enzymes. Cells can add phosphate groups to an enzyme and cause that enzyme to change shape. Cells can remove phosphate groups from an enzyme and cause an enzyme, again, to change shape. So phosphate groups are very important little molecules in living cells. ATP is really the middleman in energetic coupling. It's providing the fuel that allows cells to do their work. It's used to harvest energy from exergonic reactions and provide energy for endergonic reactions by breaking off that outermost phosphate group. And remember, there's about 7.3 kilocalories of energy, what laymen call calories, 7.3 kilocalories per mole of ATP. Organisms that use oxygen, like humans, are going to make their ATP via that long series of reactions in cellular respiration and oxidative phosphorylation. This is actually a series of oxidation reduction reactions. And in fact, anaerobic organisms can also do cellular respiration and oxidative phosphorylation, because the oxidative term refers to the oxidation reduction reactions and not to oxygen. We'll talk just a little bit about those anaerobic organisms later on in the lecture. But for now, remember that ATP is necessary for all living cells. There is no life without ATP. Let's take a minute and review the concepts behind oxidation reduction reactions. Remember, we're talking about electrons moving, being lost by some atoms, being gained by other atoms. The atom that loses an electron is oxidized, and the atom that gains is reduced. We said that there are lots of mnemonic devices out there, memory devices to help us remember which is which, but I happen to use the one Leo goes GER, and that stands for lose electron oxidation, gain electron reduction. Remember, oxidation is always coupled with reduction. In chemical reactions inside cells, if an atom is getting oxidized somewhere, another atom is getting reduced somewhere. The electron donor, in other words, the one that's losing an electron, is always paired with an electron acceptor somewhere else. Remember too that we said in oxidation reduction reactions, there are really two mechanisms at play. Sometimes the loss and gain of electrons is complete. An atom is literally losing an electron, or an atom is literally gaining an electron. But many times we're talking about a partial loss or a partial gain, and we defined that as electrons shifting position in a covalent bond. The other thing we mentioned when we were discussing oxidation reduction reactions was that every electron that gets transferred during an oxidation reduction reaction is usually accompanied by a proton, by a hydrogen ion. So molecules that contain the reduced atom often gain a hydrogen atom as well. And the opposite is also true. Molecules that are oxidized often lose a proton, along with losing an electron. So we talk about follow the proton in oxidation reduction reactions because we can identify the reduced molecule, particularly by looking for what has gained a hydrogen atom. All right, let's begin our discussion of cellular respiration now. This is a very busy slide, but it has a fairly complete summary of this process that we're going to discuss in detail. Step 1, glucose is going to be oxidized to a molecule called pyruvate. Electrons are going to be released as the glucose is broken apart. Electron carrier molecules are going to gain those electrons temporarily and hold on to them or carry them. Glucose is being oxidized. The electron carriers are being reduced. Pyruvate will then be oxidized to form a molecule called acetyl-CoA. As the pyruvate is broken apart, electrons will be released. Electron carriers will gain those electrons and become reduced. The acetyl-CoA molecule will then be oxidized to carbon dioxide. Electrons will be released. Electron carriers will be reduced. Then all of the electron carriers that are holding on to those electrons that were reduced, in other words, in steps 1 through 3, are then going to be oxidized. They are going to lose those electrons, release those electrons into the electron transport chain. The electron transport chain then is going to gain the electrons and be reduced. Electrons are passed down the electron transport chain, and their energy is harvested along the way. At the end of the electron transport chain, oxygen is present as the final electron acceptor. Remember, we are talking today about the aerobic version of cellular respiration and oxidative phosphorylation. So oxygen will be the final electron acceptor. The electron transport chain then will be oxidized and the final electron acceptor molecule will be reduced. Meanwhile, the energy that was harvested from the electrons will be used to build the proton gradient. And remember, it's this proton gradient that will then be used because energy is stored in an ion gradient. It's the energy in the proton gradient that will be used to fuel oxidative phosphorylation. This is a good place for us to finish this first video. We'll see you in the next.